Okay, so in this video, I want to show you how to do a direct stain. So direct stains are really useful in that you can easily add some dye, stain the cell, and then visualize the cell under the microscope. And so the dyes that we're going to use first, we'll use a couple different dyes on a couple of different types of cells. So we're going to first use crystal violet. Crystal violet is a basic dye. So what I want you to do is think about the PowerPoint slides, think about kind of the charge of a basic dye, the charge of the cell, and make a prediction about what you think our cells are going to look like. So I'm going to use a liquid culture of E. coli to do this. So I've already cleaned down my lab bench, everything is sterile. I'm going to take a microscope slide, and I've got down here a little setup where I can put my slide. So I will sterilize my loop, which I don't know if you can't really see it, but I'm sterilizing my loop. And then, so here's my sterile loop. Let it cool down for just a second. I'm going to take a loop full of culture and spread it on my clean glass slide. Okay, so I've got my loop full of culture. I'm going to spread that loop full of culture on my slide. And then I need to let the slide air dry. So I'm putting it back on the rack. I'm going to sterilize my loop again. And while I'm letting my slide air dry, which shouldn't take very long because I spread it out pretty quickly, I'm also going to prepare a slide from an agar culture. So when you're preparing from a liquid culture, you just dip your loop into the liquid and spread it across the plate. When you're picking from an agar, so you can see here, this is an agar slant. This is Enterococcus. So what we do when we're doing from the agar is we will take a clean glass slide again, and this time I'm going to add a drop of water onto the slide. Oop, too much water, so I'm going to dump some of that off. I just want a little boop of water on the slide. I'm going to set that down so I can re-sterilize my loop. Okay, let it cool. It's a little too hot still. Okay, and then I'm going to just take a little loop full of culture off the agar slide. Okay, and I'm going to mix that with the water I put on this slide here. So mix it, kind of spread it around. Okay. This one's probably gonna take a little bit longer to air dry, so I'll definitely like cut and paste the video. And I'm going to stain that one with a different stain so we can see what a couple of different stains look like. So I'm re-sterilizing my loop. And now I've got my original smear, which was from my liquid culture of E. coli. And I know it's really hard to see, but it looks pretty dry. So what we'll do now, our next step, is to heat fix the cells. That basically, well, it kills the cells, unfortunately, but it also helps them to adhere to the slide because we're going to be adding stains, we're going to be adding water, and so those can wash the cells away. So by heat fixing, we help attach our cells. So I'm going to pass it through the Bunsen burner. Let me scoot this over a little bit so we can see. Now, I'm not trying to fry my cells. I am not trying to cook my cells. I'm just passing through the flame. 3 or 4 times maybe. And I'm going to put that slide back now on my slide rack. I will move us over so we can do the first staining step. So with the E. coli, I'm going to go ahead and stain them with crystal violet, and so we have all of the stains at the lab bench. And so for crystal violet, let's see here, there it is. I'm going to cover the smear that I made with the stain. So here are my slides, and I'm going to add Crystal Violet so that it covers where I streak the cells. And now we wait for 1 minute. And I'm actually going to cut the minute wait out of the video, but I am currently waiting for 1 minute. So it has now been 1 minute. So what I will do is I've got my slide and I've got my tray where I'm going to collect all of my stain waste. Once I'm done doing all my staining, I will dispose of that waste in the appropriate chemical waste container. For now, I'm just going to collect it all into the tray. And so then I take my slide and I rinse with deionized water. Okay. And then what we'll do before we can visualize the slide is we will put it in some blotting paper to kind of soak up the excess liquids. And then once that's dry, we'll go ahead and visualize it. So my other smear is not quite dry yet, so we'll come back to that in just a little bit. So now our Enterococcus culture has dried on the slide. So we're gonna do kind of a similar procedure. We can now heat fix, so we will pass very carefully through the flame on the Bunsen burner. Okay, just a few times to pass through and make sure that the cells stick as much as possible. So this time I'm going to use a different stain. I'm going to use safranin. So safranin is a red dye or a pink dye. So our cells will hopefully look pink when we look at them under the microscope. Safranin only needs to be on the cells for approximately 30 seconds. So again, I'll spare you the 30 seconds. I won't make you actually watch me for 30 seconds, just sitting here and waiting. So we will again just cover the area of the slide where our cells are. Okay, so it's now been about 30 seconds. So I'm going to pick up my slide using the clothespin because I don't want stain all over me, and I will rinse off the stain very carefully. Okay. So I'm going to place my slide in the blotting paper to help it dry off. And then what I'll add at the end of the video is a picture of what the cells look like under the microscope. I'm gonna set up one more direct stain because we do have 3 that we can test. So I'm sterilizing my loop just off camera, but trust me, I am sterilizing my loop. Grabbing a slide, and I will take a loop full of bacteria once my loop cools. And I will spread that on my slide. Spread it out as much as possible so it'll air dry pretty quickly. I'm going to re-sterilize my loop. Okay. And while this is air drying, I'm going to grab my stain. And so the stain we're going to use today for this one is methylene blue. It's going to make it blue. So we're getting all the pretty colors, purple, pink, and blue. And so what the methylene blue will do, it is also a basic stain. So I want you to think about, what does a basic stain do? Is it going to stain the cell? Is it going to stain the background? So these are things that you have to think about. This culture should be Klebsiella, the genus Klebsiella. So it will be a gram negative, but we won't be able to tell, right? Rod-shaped or bacillus-shaped cell. So hopefully what we'll see after we're done staining and drying the cell is a rod-shaped blue cell because we've done just a direct stain. It looks like I have air dried pretty well, so I'm going to go ahead and heat fix by passing through the Bunsen burner. Remember, just a couple of times. 1, 2, 3. Gonna go ahead and put that on the slide rack. And the thing with methylene blue is that it actually has to stain for about 5 minutes. So of course I will pause the video and not make you watch me for 5 minutes. But I am going to add my stain to the slide and completely cover the area where I smeared the bacteria. Now I'm going to place my slide down and let it sit for 5 minutes. So our 5 minutes is up with our staining using methylene blue. So I will go ahead and of course pick up my slide and then rinse the stain away. And so you can see the stain is coming off, and we'll rinse until I've got all the stain off, and then I will put it in my blotting paper for later, and I will show you what it looks like under the microscope. So in this image, we have purple. This is our crystal violet stained slide. There is visible gram-negative rod-shaped bacteria throughout the slide. We also have some little circular pieces. Those could be debris from the media. This is a relatively old culture. It could also be a contaminant, perhaps one of our coccus-shaped organisms like Staphylococcus. But with only a direct stain, it's very difficult for us to make any sort of differentiation. All we can tell is that we stained our organisms purple, which of course was the goal. In this image, we have our direct stain of Enterococcus using safranin, which should have stained them pink. And as we can see, we do have nice pink coccus-shaped bacterium. Enterococcus is closely related to Streptococcus, which tend to grow in chains of cocci. And so you can see some of these, we've got those chains of cocci that are growing. But again, when you do a direct stain using safranin, this is the type of result you would expect to see. All of your cells should be a nice pink color. The Klebsiella stain I did with the methylene blue ended up not being as concentrated as I wanted it to be. So I redid the direct staining with methylene blue using my Enterococcus culture instead. And so what you're seeing in this image, it is a little bit blurry, I apologize for that, but you should be seeing blue stained coccus organisms. Those are our Enterococcus stained with methylene blue. What you should notice about all of these direct stains is that all of the direct stains bind directly to the cells and do not stain the background of the slide.